Asper Biotech provides genetic testing services based on Apex technology. Apex technology is based on microarray chips and is used for diseases that involve point mutations such as Noonan syndrome and cystic fibrosis. The Apex test starts with a selection of genes for PCR. The PCR is performed using uracil that enables the fragmentation of the PCR products later on. The resulting PCR products need to be purified and fragmented before the mutation analysis. This purification is done using a simple bind, wash, and elute procedure. The PCR sample is first passed through a column that binds the DNA. The reaction buffer is removed, the DNA washed and eluted from the column to obtain purified DNA. Binding the DNA to the Apex microarrays is preceded by fragmentation at the uracil sites using enzymes and heat treatment. The Apex reaction takes place on pre-printed microarrays with specially designed DNA probes. On the microarray, the fragmented DNA is hybridized with the DNA probes. Fragmented DNA and the probe on the microarray form a double-stranded DNA. Using DNA polymerase, the probe is then elongated by a single terminator nucleotide, labeled with a fluoro-4 dye. The reaction is performed with 4 different terminator nucleotides, one for each nucleotide type. Each spot on the microarray is thus labeled with a dye, specific for a nucleotide in the patient's DNA, and can be used for analyzing the DNA sequences. Detection of the fluorescent dyes on the microarray is done by using the Generama Quattro Imager platform. The Quattro Imager uses 4 different laser beams, one for each nucleotide dye, that are passed via mirrors and a prism into the microarray. In the microarray, the laser light is reflected between the surfaces where it excites the dyes. This method is called total internal reflection fluorescence. The dyes now start emitting light, and the pattern is recorded by camera and stored on a computer disk. This procedure is repeated for all 4 lasers, resulting in a complete dataset for sequence and mutation analysis. Finally, specialized software interprets the images from the Quattro Imager. All data is automatically analyzed and translated into DNA sequence information, thus revealing any mutations in the DNA of the patient. For more information, please visit our website www.asperbio.com.